Hello and welcome to the Health Tech World podcast. I'm joined today by Dr. Peter Hamley, who is Chief Scientific Officer at Samsara Therapeutics. Thank you very much for joining us, Peter.
Thank you, Gordon. Thanks for inviting me.
So I've asked Peter in today really to talk about a couple of exciting developments in the world of Alzheimer's research. In July, you may have seen there was a flurry of news stories about a new drug called donanemab, which was shown to slow the pace of Alzheimer's research of Alzheimer's disease by around a third. And the drug works by clearing a protein that builds up in the brain of people who have Alzheimer's disease specifically. However, there have since been a number of articles and opinion pieces urging caution about findings. Peter, have you read much about this? What are your thoughts on the research that you've seen yourself?
Well, I think donanemab and lecanemab are real advances, real medical advances. I mean, certainly they have an effect on slowing the progress of this really complex and terrible disease. So, you know, that's something that we should be really pleased about, particularly in the scientific community. And what I think is particularly exciting is that it's a validation of all the research that's gone into the concept behind these drugs over the last probably 20 or more years. So that's very good. For patients, it's not really a breakthrough, it's a help for them, potentially a few more months of clarity, I guess. But it's not really a game changer for patients. And then, of course, there are some other aspects that have to be considered, like the potential side effects that have been observed in clinical trials. So it's good news for patients, but not, you know, the breakthrough that everyone's looking for. But on the other hand, the science behind it gives us an opportunity to really progress and get some better things out there.
I think it's just one of those, obviously it's one of those issues that it just affects so many people, obviously the people who are themselves impacted by it and all their families. So I can see why people can kind of get quite carried away by what looks like quite exciting research, but obviously there's so much more to it. It's not necessarily as straightforward as it may initially seem.
Yeah, I mean, Alzheimer's is a sort of multifactorial disease. There's so many potential factors that go into, you know, the origin of the disease, and I think people have really struggled, or scientists have really struggled over the decades to unravel all this. And I think the way that, and we can talk about that later, but the way that these new drugs work is very specific. It's going after one specific thing. It's pretty unlikely that Alzheimer's is just driven by one factor. So I think there's got to be a lot more work going into this before we get a true breakthrough drug that will really halt and ideally reverse the disease. But there is a lot going on, and this is a great start.
Yeah, so that kind of leads us nicely really into the work that you guys are doing at Samsara in developing a new range of therapies that are designed to either prevent or halt the progression of various neurodegenerative diseases, including Alzheimer's, by boosting a process called autophagy. Could you just tell me a little bit about what that is and how this could potentially help to tackle Alzheimer's disease?
Yeah, very glad to. And I would say that it's part of the same story, but hopefully it's an evolution of that. So autophagy is a really interesting process. It's very fundamental. It goes on in all the cells of your body all the time, and it's sometimes described as the cell's waste disposal and recycling system. You can also call it the cell's quality control system. So what it's there for is to get rid of unwanted debris that accumulates in your cells, and that can actually be a number of different things, including bacteria and viruses, but the most important one really is proteins. So proteins, when things go wrong in your cell, proteins can, and not just go wrong, but when they reach their sell-by date, proteins are no longer required, and there needs to be a mechanism for getting rid of them. And so the cell's main mechanism for doing that is the process of autophagy. So what happens is the proteins that need to be removed are marked for so-called degradation, and then there's a really interesting mechanism, very complex mechanism, that creates a membrane structure around this protein debris, and then that structure, which is actually literally a sphere, so you can visualize it using microscopes as little spherical objects. These get taken to the lysosome. The lysosome is the cell's degradation machinery. So that's where everything gets pulled apart and recycled. So that's the process. But you can imagine there's a number of different steps in that process, and it's a pretty complex one. So autophagy, as I said, it happens all the time in your body, and in times of cell stress, autophagy, the level of autophagy goes up. So that whole process of recycling those unwanted proteins. And a cell stress could be, for example, starvation. So it's been shown in many different organisms, even in humans, that if you starve yourself, for example, intermittent fasting, which is a very popular activity these days, you can actually measure the levels of autophagy going up in human cells, which is a good thing, right? Because autophagy is the way that the cell refreshes, renews itself. It's also been shown in heavy exercise. So if you exercise and then you look at your muscle tissue, you can see levels of autophagy going up there. So it's kind of associated with good, healthy things. And there's a couple of other things before I get onto the connection with Alzheimer's. So firstly, there's a lot of evidence that if you can have good levels and maintain good levels of autophagy throughout your life, and this is obviously not done in humans yet, but in organisms such as mice, up to mice, you can see a very significant increase in longevity, so lifespan. And it's not so much lifespan, but what we're interested in is health span. So I think the figure for mice is something like 35% longer living mice if they have boosted autophagy. But the mice are also a lot healthier. So, you know, their fur doesn't disappear as they get older, and they're bounding around with more energy. And of course, that's really something that would be great if we could do that for humans. So those are all the positive things about autophagy. It's a really important cellular process. The problems start when autophagy goes wrong, and there's a absolutely enormous amount of scientific literature showing that this is the case, that in many diseases, autophagy doesn't work as well as it should, or it doesn't work at all, and that causes many different diseases. So that means that it's been a really growing area of medical research, particularly I'd say in the last 10 years. So the Nobel Prize was awarded in 2016 to Professor Osumi in Japan for working out some of the mechanisms that underlie autophagy. And since then, there's been a lot more interest in it. So when autophagy goes wrong, you can imagine the cell doesn't recycle properly, it doesn't renew itself properly, and toxic proteins can build up. That's where we come to neurodegeneration, in particular Alzheimer's, but also Parkinson's disease, motor neuron disease, and many others. And the hallmark of these diseases may not be the cause, and as I said, these are multifactorial diseases because they're so complex, but the hallmark that you can see in all these diseases is a buildup of toxic aggregated proteins that shouldn't be there. The classic one that a lot of people have heard of for Alzheimer's disease is called beta amyloid. So that's a protein that gets sort of excreted from your cells, and you can actually observe these so-called plaques in the brains of Alzheimer's patients. And the big question is, are those plaques causing Alzheimer's disease, or are they a symptom of the process?
Yeah.
And that's a question that no one has really got a definitive answer to even now, although I think these new drugs may give us a hint here. So basically, autophagy is the body's process for removing those protein aggregates. And we've shown, not just we, but many groups have shown that in Alzheimer's disease, in Parkinson's disease, in motor neuron disease, in Huntington disease, these toxic protein aggregates are associated with a lack of autophagy. So autophagy is broken when you have those aggregates. And sometimes we call that a sort of vicious cycle, because the more toxic protein you have, the more your autophagy system goes wrong.
Yeah.
And the less autophagy you have, the more protein builds up. So it goes round and round. So really you need to break that cycle. So what we want to do in Samsara Therapeutics, which is a company that is really the leader in autophagy-based medicines, we're looking to make a therapeutic that induces autophagy in the brain. If you can induce autophagy, you should be able to remove these toxic proteins. And then that comes back to how do these new drugs work? They don't specifically look at autophagy. What they do is just simply go after those aggregated proteins. In the case of the two you mentioned, they go after beta amyloid and they remove it. And we can see from the results that they do have an impact on the progression of the disease, albeit not very huge. So, and this is my final point, so hopefully you've understood what I said. The idea of boosting autophagy is we will not only remove the aggregated protein, for example, beta amyloid, but we will also restore the lost autophagy to the cells. And hopefully you've convinced now that autophagy is extremely important to maintain the health of the cell. So we'll not just remove the toxic protein, but the cellular health will be at least partially restored. So we think this approach it's connected to the approach that's already shown to be working, but we think it's going to add an extra dimension of hopefully a positive therapeutic outcome. So that's why we're so excited about what we're doing.
So I guess it's just the same process then for different conditions. Obviously, the drug might be slightly different, but they're still going to be following the same process within the brain.
Yeah. So maybe I didn't say that. There's a different protein aggregates formed for different neurodegeneration diseases. Actually, sometimes in different parts of the brain, but the principle is the same. So we are actually, if anything, more interested in Parkinson's and motor neuron disease, which is also known as ALS, in Samsara. We've got some nice results in Alzheimer's, but Parkinson's is the protein that is the hallmark of Parkinson's is called alpha-synuclein, and that forms inside the cells in your dopaminergic neurons. You might know that Parkinson's is a disease where your ability to produce dopamine reduces, and that's where you get the terrible motor symptoms like the tremors. So alpha-synuclein builds up in those neurons, and we've shown in animal models and in patient cells that we can remove the alpha-synuclein just by boosting the levels of autophagy. And just to complete that, ALS, or motor neuron disease, which is an absolutely awful disease with no real good treatments, that appears to be driven by another aggregated protein, which is called TDP43. And again, we've shown that boosting autophagy in animal models and in patient motor neurons, because motor neurons are the ones that are responsible for ALS, reduces the amount of TDP43. So it's all kind of the same approach. They're similar but different diseases. And anything that works in, say, Parkinson's disease, that's an autophagy inducer, will also work in Alzheimer's. And we've got some data to prove that as well.
So you're working with the Michael J. Fox Foundation, is that correct, with that with the research that you're doing?
Yeah, so yes, we're working with them for our second project, which is conveniently called SAM002. That's primarily at the moment for Parkinson's disease, but we're also testing that in Alzheimer's at the moment. And then SAM001, which is our most advanced program, hopefully should be going into a clinical trial very soon, in the next few months. And that is currently firstly targeted to motor neuron disease, but we very strongly believe that this can be used for Alzheimer's and Parkinson's as well.
What makes it so challenging to find treatments for these conditions? Because as you say, there's so few treatments that do, that even slow the progression of the disease. What is it about them that makes them so hard to treat?
Well, as I said, first of all, it's the complexity of the disease. So, if you want to come up with a drug, you need to know what's causing the disease. And unfortunately, with these diseases, they are very complex. There's a lot of controversy about risk factors, for example, in these diseases. For example, Parkinson's is often thought to be largely created by sort of outside influences like pollution, for example, or potentially brain injury. But that's very much not really proven. So, and then we know that the mechanisms are very complex and multifactorial. So that's one thing. And the other thing is that they are brain diseases. So that's a massive challenge for drug discovery and technology, because you've got to be able to get your molecule into the brain, and the brain is built to keep stuff out of it, obviously. So it's much easier to get drug into any other organ. I mean, it's still not easy, because drug discovery has been said to be one of the hardest possible things you can do, probably harder than landing a man on the moon, because that's a technical challenge. Whereas in drug discovery, it's a mixture of all sorts of challenges. I could go on forever about that. But to get it into the brain, you need to get past the blood-brain barrier, which is designed to keep foreign bodies out. Some people have managed to do that simply by injecting straight into the spinal cord, but that is not very pleasant for patients, of course. And even then, it's not a very efficient way of getting to the site of action. We're doing a more classical way, which is using small molecules, so most marketed drugs are small molecules. And the advantage of them is that they do get into all sorts of places in the body, but it's still very difficult to get them into the brain, and you have to make sure that when they're in the brain, that they are actually free to bind to your target, because they do have a habit of binding to other proteins as well. So it's quite a technical challenge to get any drug for a neuronal indication, unfortunately.
Well, it sounds like an exciting field to be part of, and this research sounds like it could be incredibly promising. So what do you have coming up for the remainder of 2023 and 2024? What's the sort of timeline looking like?
Well, so for SAM001, which is as I said, a very exciting molecule with a completely novel mechanism of action, we are in late stage toxicology, which is what you need to get the next permission, basically, for the next step, which is to put it into human volunteers. In order to do that, you really have to prove that your drug is something that's safe and can be given to humans, to patients, without any untoward side effects. So that requires a lot of animal testing, unfortunately. Hopefully we reduced it somewhat in the last few years, but it still requires that. And a lot of other measurements that need to be taken on it. So far, touch wood, things are going very well for SAM1, and so, as I said, the idea is we get it into humans, hopefully sometime next year, certainly in the first half of next year. Meanwhile, we're doing all sorts of other things in Samsara, because we think the whole area of autophagy is really going to be ripe for coming up with lots of different types of drug. So just to go back to autophagy, it's really interesting because it's such a complex process, and that gives us a whole load of opportunities to find new biology. So, you know, it's kind of the whole thing is orchestrated by a sort of multitude of different mechanisms and proteins all acting in concert together. And one of the things that we are good at in Samsara is identifying some of those new processes which are new, and that allows us to create design drugs that are really quite special and new. So SAM002, which I mentioned already, we're still working on that to try to get it to get that one molecule that we need to then put into the toxicology that SAM1 is in, with the idea that hopefully in a year, 18 months, we can get that into patients as well. It's got a completely different mechanism. They both induce autophagy, but in a completely different way. So there may be some real advantages between the two. Maybe one is better for one disease and the other is better for another. And then behind that, we've got all sorts of earlier projects going on. We've got a new molecule which we're currently looking at for completely different diseases. So fibrosis, for example, which is all about wound healing and things like that, which is where autophagy is also heavily implicated. But we're actually spoiled for choice in terms of where we go next, because autophagy is, as I said, involved in so many different diseases.
Brilliant. Well, thank you so much for taking the time to talk to me today. Really excited to see where things go from here. So we'll be sure to stay in touch and maybe catch up with you again in a year or so once things have progressed a bit.
Great. Happy to.
Brilliant. Well, thanks a lot, and enjoy the rest of your day.